All right, welcome, ladies and gentlemen. We're going to talk about phylogeny today, which, if you happen to be a fan of The Simpsons at any point, is the source of this lovely tree of life right here. Isn't it just gorgeous? Let's go ahead and let's start getting into things. We're going to talk primarily about phylogenetic trees and cladograms, and these are diagrams that are used to show evolutionary relationships. You may not have seen one kind of like this one yet, although I do have a necklace that I occasionally wear which has a simplified version of this. But this image is showing a phylogenetic tree showing the relationships of all the organisms on the planet to each other. Apologies, starts over here with Luca, the last universal common ancestor that we talked about in a previous lesson. And then you have the first organisms that kind of branched off of that would have been the archaea and the eubacteria. And then what actually happened is the archaea are thought to be the ancestors of all modern eukaryotes. However, they did that by doing endosymbiosis or taking in a eubacterial ancestor, and so it's kind of weird. It's like archaea and eubacteria had a baby, and that made eukaryotes. So phylogeny is just the term we use for looking at the history of these various evolutionary relationships. We can look at a phylogenetic tree of every single organism on the planet, which is what we see here, although you don't see every single species represented. We can look at the relationship between major evolutionary groups like plants, animals, fungi, protists. We can look at small groups of closely related species. We'll look at plenty of phylogenetic trees and cladograms of those. We can look at relationships between populations. We can even look at kind of divergence in genes between common ancestors. So, what is a phylogenetic tree? Well, a phylogenetic tree is basically a diagram that's showing you evolutionary history. And that's going to be different from the other type of major diagram that we're going to talk about because in this one, distances are important. It's kind of like when you're graphing something, if you had a time axis and you were graphing something along it, that would give you a unit to measure. In a phylogenetic tree, there is always an implied time axis to it. And so as you go further on from the root of whatever that phylogenetic tree is, because they always start in a root and end in some final species, the root is usually going to be farthest back in time, and where you have more species is going to be the more recent time, and going from farther back in time to more recent in time, that's going to give you your time axis. It's not necessarily going to be explicitly drawn on all of these, but the understanding is usually the root is the furthest back in time. If it's a phylogenetic tree, it's usually going to be based on something more than just morphology and how, how fossils look. It's probably going to have some absolute dating or relative dating to signal around how many millions of years or thousands of years have passed since you had a common ancestor between two organisms. And the most accurate phylogenetic trees are going to use molecular evidence. So biochemical pathways, DNA and protein sequences, and molecular clocks, which we'll talk about any of those that we haven't talked about yet. So what is the molecular clock, since that's important for developing these phylogenetic trees? Basically, it's the idea that you have in a common ancestral species an ancestral gene that would have had very similar sequences for every member of the population. However, mutations will build up in genes, and even the most important of genes that you try to keep mutation free because they're so essential for functioning of cells, even those can build up mutations. They just may do so at a very slow rate. So in this image on the top, what we're seeing is a situation where this gene accumulates one mutation every 25 million years. So that's one nucleotide change every 25 million years, which means if this diverges into two different groups of organisms, after 25 million years, on average, you would expect to find one difference in in a nucleic acid sequence from the original ancestral gene. Then another 25 million years passes, and you can accumulate a second mutation. So now instead of having one nucleic acid difference, you've got two nucleic acid differences. Or twice as many nucleic acid differences. So the theory behind this is if you can measure how many differences there are in a sequence of DNA or in an amino acid sequence, then you can gauge approximately how many millions of years ago those two organisms that you're looking at shared a common ancestor. So, for example, there's been some work done to calculate this for several genes found in various organisms. For example, the alpha globin part of a hemoglobin molecule seems to experience base changes at a rate of about 0.56 bases per billion years. So you're not going to be able to see like super rapid evolution when you're looking at this, but you'll be able to see, okay, well, what's the relationship between organisms that may have had a common ancestor about 1 billion years ago. Now, this does depend on kind of going back and kind of providing dates for this molecular clock. So to calculate how fast DNA changes, you do need to be able to use relative and absolute dating techniques to figure out the ages of various species. And using that, you can calculate the rate of mutation. And using the rate of mutation, you can use the number of mutations or differences, and you can extrapolate from that the amount of years it's been since organisms shared a common ancestor. So here on the bottom we have, looking at a particular protein, the proportion of amino acid differences. So basically the higher this number goes, the more amino acids are different in a particular sequence. And as time passes, you start to see this very strong linear pattern of an increase in the number of mutations as millions of years pass. And that's the molecular clock that you can use for phylogenetic analysis. A cladogram is something different. While it shows relationships, it does not depict amount of time, genetic distance, or evolutionary change. All it's showing is that things are different. A lot of times it's showing you clades, and a clade is basically a group of ancestors, a group of organisms that all come from the same common ancestor. So you could look at a clade of Dinosauria, which is every organism coming off of this location right here. You could look at the clade on this cladogram of Saurischia or Ornithischia. And those are three different clades represented here, and there's probably a clade back here, maybe like Diapsid, that includes pterosaurs as well. Notice you have no sense of time here. You don't know whether Ornithischia and Saurischia branched off into their different species at the same time. For all you know, Saurischia could have happened like closer to the split between the split within the Saurischians could have happened much closer to the split amongst the dinosaurs. It's not really shown on this. This is usually formed on the basis of morphological characteristics, kind of like what we have listed here. So for example, if you're a dinosaur, you have upright posture, bipedal locomotion, you live on land, your vertebrae are regionalized, you don't have very large fourth and fifth digits, meaning your fingers, and you have a small size, and you do have an opposable thumb. So it's usually based on these morphological characters. So a lot of fossil evidence is used to construct cladograms. Okay. So what's the major difference? The major difference is that phylogenetic trees show time. That's about it. They either depict time or the amount of difference between two populations or two sequences. They are going to give you some sort of degree of how fast things are going or how closely related two things are. So, for example, here we have 4 species, A, B, C, and D, and we're showing the relationships in a cladogram and a phylogram. You can get the idea from both of these that A is more closely related to B, and C is more closely related to D, because they share the more recent common ancestor. Whereas if you're trying to look between species A and species C, they have another common ancestor even farther back, so they're probably less closely related. If you look at the phylogram, though, which is another name for a phylogenetic tree, so let's just put that there so that way we remember it. You'll see that there's this kind of difference in this distance, which is showing you when the common ancestor of A and B was around, and this distance, which is showing you when the common ancestor of C and D are around. So you should expect to see from this a few things. You should expect to see that C and D might have more similarities, whether they're genetic or whether they're morphological, than A and B do. Why? Because their common ancestor was more recent in history, so they haven't had as much time as species A and B have to accumulate differences. So that's the kind of difference that you can get from these types of diagrams. Usually you'll be told whether you're looking at a cladogram or at a phylogenetic tree. So let's look at how you construct these. You can use morphology, basically how something looks, how the body parts work and fit together, which is sometimes all we have, especially for fossil evidence. But if you have access to molecular data, it's a lot more accurate and a lot more reliable. So one thing you can do is you can build a phylogenetic tree based on what traits were gained or lost. And this is done by analyzing characters. A character is just a certain body part or property of an organism that you can trace to try and figure out, is it in all the descendants? Was it in the ancestor? So this can, the trait that was in an original ancestor is called the ancestral trait, and the trait that is in the descendant is called the derived trait. So a trait can be ancestral or derived depending on what you're looking at. So for example, feathers are an ancestral trait for all modern birds, meaning the common ancestor of all modern birds likely had feathers, and that you are, going to see that in all the descendants. However, when you're looking at all living organisms or all living vertebrates, they end up being a derived trait that's found only in small parts of the population. Another example is hair. Hair is an ancestral trait for all mammals. Since we can theorize that the ancestor of all modern mammals had hair, even in groups of mammals that don't currently have hair, we can kind of link their relationships to us. So for example, whales don't have a lot of visible body hair, yet they are still mammals because of other characteristics, and from that we can say, okay, they lost this ancestral trait. Similar things for other aquatic mammals as well. However, our hair is a derived trait that is different from our common ancestor with birds and reptiles and everybody else. So, shared traits or derived traits show evidence of common ancestry. So, for example, here we see a phylogenetic tree that is showing you the relationships between different vertebrates. No invertebrates are on here, as you can see. And so shared or derived characters are usually found in more than one organism, more than one lineage. So the shared or derived character for every organism on this picture right here on this phylogenetic tree is we all have backbones. We all had notochords, an undivided supporting rod. We are all vertebrates. So that is the shared or derived character for all of the organisms on this slide right here. When we're looking at phylogenetic trees, a lot of times what we're going to see is we are often going to see what is called an outgroup. An outgroup is kind of like your comparison that you're comparing all these related organisms to. It's going to be the most distantly related, so it's going to end up being the most different from the rest of the other organisms. So on here, you'll see a phylogenetic tree of various animals. And the outgroup here is the one that shares the common ancestor that's most distant. So for example, between all of these groups, the common ancestor that's farthest back here is the one that looks like the common ancestor to all animals. And notice that there is one species there, one group that branches off here, Porifera, the sponges, and their common ancestor with all the other animals is much farther back in time. whereas, for example, Annelida and Arthropoda have a fairly recent common ancestor. Annelids are worms, arthropods are insects. So sponges are the outgroup in this cladogram. In this cladogram right here, the outgroup is hagfish. Why? Because their common ancestor with anyone else is the furthest back in time. They are the least related to everyone else. So in a lot of the cladograms or phylogenetic trees you will see on the AP test, they're likely to be an outgroup in there somewhere. So when we're drawing these, we usually have our ancestral species, and then we draw a line showing its link to its descendant populations, and that's our lineage that we talk about. Anytime we have a split, or a node, if you want to call it that, is where you have one species divide into two. So that split, that node, represents the most recent common ancestor of two groups of organisms. It could be between two species, two genera. It could be between two individual populations. If you're showing species, this could be a speciation event, basically showing you where a common ancestor diverged into two different groups that would end up becoming two different species. And then the common ancestor of all the organisms in a tree is going to form its root, which is going to show, the common ancestry of everyone. So here we've got a little mini primate phylogenetic tree. We can see that orangutans are the outgroup because they're the most distantly related. The two organisms on here who seem to share the closest relationship or the most recent common ancestor would be here, chimpanzees and humans, which had a common ancestor around 5 million years ago, whereas the common ancestor of all four organisms on here probably was around maybe 13 million years ago or so. The vertical distances between these branches usually don't play a role unless you're told otherwise. And then the vertical order when you get to these end branches really doesn't matter. So for example, I could have chimpanzee here and human here, wouldn't matter. I could have human here and chimpanzee here, wouldn't matter, because either way, we're seeing the same information on this chart, that humans and chimpanzees diverged from a common ancestor. We cannot, however, stick like a gorilla in here. Why? Because that would kind of disrupt the relationships we're trying to show. Gorillas and humans are not more closely related than humans and chimpanzees. So humans and chimpanzees need to have that same branching off point. It'd be like if this was a physical structure and you can rotate these guys. I could even rotate around here, or I could rotate around here. That's fine. The only thing I can't do is I can't like switch this guy to another branching event. So a clade, which is, where the word cladogram gets its name from, is all the evolutionary descendants of a common ancestor. So if we trace back the common ancestor of a lot of these vertebrates, then that would mean that all of us who have vertebral columns are in the same clade because we all share that common ancestor. What you could do is you could pick some other point and you could say, okay, well, I am going to say trace from this point. Where did my cursor go? There it is. I could trace from this point on, and every organism from this point on, lizards, crocodiles, pigeons, mice, and chimpanzees, are all amniotes. So that's the clade we're looking at because they are all the descendants of a common ancestor. Two clades that are closely related to each other or are each other's closest relatives are what are called sister clades, and two species that are each other's closest relationships are called sister species. So for example, not here, but here, humans and chimpanzees, we're both individual species. We are each other's closest living relatives, so we would be sister species. Though we're not as quite as closely related as we would be if we, say, had some Homo neanderthalis, Neanderthalensis, sorry, brain tongue fart, lying around. Now, phylogenetic trees and cladograms don't stay static. they change based on new evidence. They are just hypotheses that need to be backed up with evidence, backed up with data. So as we uncover new fossils, or as has happened in the last few decades, as we get better at analyzing genetic data and molecular data, we've had to revise a lot of phylogeny. So here's one example of that. Pay attention to where whales are on this. So whales are mammals. Whales are descended from terrestrial mammals, meaning their ancestors used to live on land. And for quite some time, it was a mystery, where did these guys come from? So who was their closest modern living relative on land? And for a while, it was thought to be this group of now extinct organisms called mesenchids. However, using molecular evidence, it's been shown that mesenchids would actually be the outgroup here, and that hippos and whales share the closest genetic relationship. So a whale is more closely related to a hippo than it is to, say, a cow or a goat or a camel. So that's going to be its closest living terrestrial relative. But then if we get further evidence at some point in the future that doesn't support this phylogenetic tree, we can modify it in the future based on the new evidence. So let's try our hand at constructing a phylogeny. So one thing that you might see on the AP test is a kind of list of traits like this, where they'll show you a plus when an organism has a trait, and a minus when it doesn't. So if you look at this, you can see that, for example, pigeons have jaws, lungs, claws or nails, gizzards, and feathers. They do not have fur or mammary glands, and they do have keratinous scales. So you can look at these traits and you can actually use them to try to create a phylogenetic tree, or in this case a cladogram, since we don't have any time or degree of divergence data. So what you can see is, okay, the lamprey was always the outgroup because it doesn't have any of these traits. All right. You can also see something interesting with keratinous scales. There's only these three guys that have it, so it needs to only be present on a branch that leads to lizards, crocodiles, and pigeons, but not to mice and chimpanzees. And you can see that divergence here, where on one branch you went on to have fur and mammary glands, and the other you went on to have keratinous scales. Okie dokie. So now we're going to try and do that ourselves with some other characters. So what we see, and what you can do when you're drawing one of these, is you can just kind of, if you're doing this on the test, they will have a diagram for you that you need to fill in. So you won't need to decide about where things branch as much as where the modern species eventually end up. But if you have to draw one of these from scratch, you can start out with one line that ends up leading out to your outgroup or outlier, and that would be your hagfish. Why? Because they only have one of the 6 traits listed here, a heart with chambers. Everything on this has to have a heart with chambers. Why? Because every organism on the list has a heart with chambers. If you look at the next trait, which is jaws, only these guys have jaws. So you need a branch that splits off after a heart with chambers, because you know everything after this point has a heart with chambers. No, it needs to split off, and it needs to have, let's see, let me do this a different way to make this easier. Apologies, it's been a while since I've drawn a cladogram from one of these things of traits. So we're going to have splitting off after the heart with chambers. Let's have the hagfish. The next trait on the list is actually jaws. Okay. Is there anything that has jaws but does not have the next trait of bony skeleton? Yes, it's the shark. So the shark is going to split off. Next trait is bony skeleton. And we have an organism that has a heart with chambers, jaws, and a bony skeleton but lacks the next trait. So it's going to branch off, and that is going to be our toad. Okay, the next trait is going to be an amniotic egg. Do you need to know what each of these traits are? Not necessarily. Would you probably be interested in knowing what these traits are? Maybe. You could always look them up. All right, so snakes have an amniotic egg. They do not have the next trait, which is hair. Then an organism that has hair but does not have the next trait is the spiny anteater. And then your next trait is a placenta. And the organism that has that and all the other traces would be the horse. So that is a very simple way of showing this. Notice I don't have any time scale. I don't know how physically different these organisms are from each other. I don't know how much genetic difference they have from each other. I'm listing them out by traits, so I'm making a cladogram. So how do we use these in science? Well, what we can do is we can use these cladograms to try and figure out not just how organisms are related, but also to try and figure out events that happened in the past. So, for example, with zoonotic diseases, you can try and create a cladogram to show, okay, humans acquired this disease from another animal. What other similar diseases in other animals is this virus or whatever it is that we're looking at most closely related to? So on this chart, what we're looking at is the ancestry of two major strains of HIV, HIV-1 and HIV-2, both of which infect humans. However, when you look at them and look at their genetic similarities, what you find, or what scientists found, is that there is a simian immunodeficiency virus, the monkey and primate equivalent, which is found in chimpanzees, which is very closely related to HIV-1. But it looks like there's a second strain of SIV in Sudi mangabes that's more closely related to HIV-2. So even though you might probably have heard rumors that HIV came from some sort of interaction with chimpanzees, it turns out there are multiple strains, and humans actually got infected with HIV at least twice. Once from chimpanzees and once from Sudi mangabes. Then what can happen is, so there were a lot of social and political issues around the development of HIV in the 80s, and people were trying to trace where it came from when it arose, because they were trying to blame one group or another, or prevent blame from being shifted onto one group or another. And so they traced the ancestry of all these various strains of HIV that had infected different groups of people. And what they found by using molecular clock data is that HIV seemed to have entered the human population from chimpanzees, at least HIV-1 did, back before around 1930. Because notice, so this is showing branch length from a common ancestor. So it's showing you degree of difference. And then we have these different variants. The earliest date we had on here was 1959. But, it shared, this family of HIV shared a common ancestor with this family of HIV, which happened far enough back in time, and they could estimate that that would have been around 1930. The thought is that the infection probably happened from a human eating bushmeat, which is chimpanzee meat, that was not properly cooked. It can also be used to try and figure out some ancestral characteristics. So one thing that you might have noted is that if you had a trait that is in all the descendants of a common ancestor, like let's say a trait that you find in the green annel, zebrafinch, chicken, and the American alligator, and then you find some other related organism that's, say, related to the American alligator that doesn't have this trait, you can probably infer that, oh, its ancestor had that trait and it lost it. It can also lets you draw conclusions about extinct organisms. There's a lot of information we can get from the fossil record. Unfortunately, there's a lot of behavioral evidence and information that we just can't get. So one thing that was done was opsin proteins, which are some of the pigments involved in vision, were reconstructed looking at the opsin proteins in a group of related organisms. So this is the ancestor of Archosaurs. And so what they could do is by looking at these opsin proteins in a green animal, a zebrafinch, a chicken, a domestic pigeon, and an American alligator, they could try to reconstruct the approximate structure of that opsin protein. And from that, they were able to infer that, this archosaur way back in the past was probably nocturnal. Because depending on whether you have a lot of rods or rods and cones, you'll either be a better nocturnal viewer or you'll be better at viewing things in full daylight. And that is it. Next up will be population genetics, which hopefully will follow in a couple of days.